Speaker is Ryan Schenck. Polyclonal path toward malignant transformation in familial adenomatous.
All right. So we'll go ahead and continue talking about pre-malignant biology with my talk today. So as Bob said, I'm going to be talking about the polyclonal path to malignant transformation in familial adenomatous polyposis. So there's still a lot of unanswered questions about tumor initiation. We've done a great job since the advent of next generation sequencing of trying to characterize the genomic landscape of cancer through TCGA, PCOG, numerous other initiatives. And more recently, we've really begun to understand that cancer driver mutations are present pretty ubiquitously through histologically normal tissue and are competing within that normal tissue throughout our lives. And this really creates a problem where we don't have a good understanding of how we get from that histologically normal tissue with numerous cancer driving mutations towards that malignant state. And these questions really revolve around the growth dynamics and the loss of homeostasis leading to tumorigenesis, and whether these cancers, these malignancies are emerging from a mono or polyclonal initiation route, what the timing of that tumorigenesis is, and the changes in selection pressures over time. And the initiatives such as the Precancer Atlas are really trying to help us bridge that understanding between this normal tissue and cancer. So what I'm really focused on is the familial adenomatous polyposis, or FAP for short. We believe that it's a really great model disease to understand pre-malignant biology and initiation. FAP is a predisposition to colorectal cancer due to a germline APC mutation. So these patients just have to get that second hit, which tends to kind of set off that initial adenomatous polyp and the hyperproliferation. And these patients present with hundreds to thousands of polyps that are spanning this pre-malignant stage between the benign adenomatous polyposis polyps to the malignant dysplasia and adenocarcinomas. And by the time these patients are in their mid-30s, they often receive a total colectomy when resection is no longer possible and you can't keep up with the polyp burden. So this creates a lot of tissue that we can sample from to be able to study these different pre-malignant states. So our primary cohort consists of six patients where we have 150 different whole genome or whole exome samples from independent polyps spanning the entire colon. And then we have a couple of other cohorts that we're using as comparators, one being a sporadic colorectal cancer cohort that utilizes a different sampling technique. So here they're using a multi-region of a single polyp. Very rarely do synchronous sporadic colorectal cancer polyps emerge. And then another cohort that also uses a multi-region approach that's also an FAP cohort. So I'm not going to bore you with talking about the genomic landscape because I don't think that many of you will be too interested, and I don't want to talk about it. But what I do want to say is just that the FAP genomic landscape kind of mirrors that with what we see in the sporadic colorectal cancer, where we see benign to adenocarcinomas increasing the somatic alterations that are present. So what we're really interested in is the clonal evolution of these pre-malignant tissues to the malignant polyps. So the benign samples tend to exhibit higher heterogeneity. They're more subclonally dominant. The dysplastic samples that we have are going to be more clonal. And that kind of mirrors what we're seeing in the sporadic colorectal cancer cases, where we see higher evidence of more clonal mutations compared to the benign samples. And when we run DN over DS to kind of look at evidence of positive selection, what we see is that the benign polyps, as well as in the sporadic cases, the adenomas have higher evidence of positive selection compared to the dysplastic adenocarcinomas and the carcinomas in the sporadic cases. So we also use a Bradley-Terry timing analysis to try to look at the preferential ordering of these driving mutations. And we see that in the benign samples, they always have APC preferentially early as that first driver event. And then the late events tend to be the KRAS, TP53, which dovetails nicely with the dysplastic samples where we see those early events, those that are being late in the benign tend to emerge as being early in our dysplasia and adenocarcinomas. So any study of clonal evolution isn't complete without some phylogenies. But first I'm gonna kind of walk you through the unexpected results that we got when we tried to create these phylogenies of our samples by showing you some multi-region sporadic results. So this is a single carcinoma, which has 4 different samples taken from a single polyp. You can see the consensus tree there, which is just the majority rule tree, showing pretty good branch support, indicating that we have high confidence in being able to resolve this evolutionary history through this phylogeny, with a pretty low homoplasia index, which just is telling us how well characters or mutations fit on this tree. When we did this for our samples of these independent polyps, we got a very different result. We had a very hard time being able to determine the majority consensus of this tree. We see that samples don't clade very well, and they don't clade by colon region or polyp type, and they had really high homoplasia index metric. And that was pretty consistent for all of our patients. So all of our multi-lesion patient samples exhibited very high homoplasia compared to the multi-region, which consistently has much lower homoplasia for both the sporadic and the FAP. So this is kind of interesting and strange. So we took a step back and we want to just kind of compare on a patient pairwise basis what these mutations are that are contributing to the difficulties of resolving these phylogenies. And I'm going to walk you through kind of how we got there and go into the weeds a little bit. So the first step is we construct these posterior probabilities for the purity and ploidy adjusted variable frequencies. So this is commonly referred to as the cancer cell fraction. And this is for each of the mutations across all of our samples. And here I'm showing in these joy plots the posterior distributions for APC mutations. And what you can see is at the top, those distributions that are shifted towards the left or the lower frequency, those are going to be our high confidence subclonal mutations. And those that are closest to the bottom are our high confidence clonal mutations. For APC, we see that high confidence clonal distribution increase in our dysplasia samples, and those that are towards the top are in the more benign samples. And we can visualize these posteriors using these plots in the middle here. So that histogram there is what you're usually used to seeing for these variable frequency or CCF distributions. But we can also combine that with the probability of those mutations existing in what we would consider to be a clonal frequency. And then, so using this information, and that lower one there actually is showing evidence of two subclonal APC mutations within a single sample, which is quite interesting. So we take all this information and we make a pairwise comparison of these mutations so that we can see what extent of sharing is present between each of the samples across within each of our patients. And we color these by the shared probability of one of the mutations existing within the clonal range. So when we make these comparisons, what we observe is there's extensive subclonal sharing between samples of all types, but with a much higher degree of sharing between our benign samples. And we don't see any correlation between physical distance and similarity. The similarity metric that we're using is the weighted cosine similarity, which is we're weighting by the probability of existing as a clonal mutation. So if you have really high subclonal shared mutations, you're going to end up with a really high similarity score. But what we see is that the benign samples tend to exhibit a higher degree of sharing, while our benign versus dysplastic and dysplasia samples against one another they still exhibit sharing, but it's much less than our benign samples. And so we wanted to characterize what are these subclonal mutations that we're observing. And a lot of them are exhibiting early clock-like mutational signatures of normal cell turnover. And very interestingly, we see that about 50% of these shared mutations are at least one colon region apart. So mutations that are shared between the ascending colon and the descending, but not in the transverse in the middle there. And that's a pretty big fraction, and it's also a very large distance to have these shared mutations. And so we think what's going on here can be explained through a conceptual model of polyclonal initiation. So we're thinking that during early colon development, during embryogenesis, you're beginning to accrue somatic mutations as you get that epithelial sheet that's beginning to form prior to the crypt formation. And then in the middle of kind of embryogenesis, I think in mice it's week 15, 11 to 15, you begin to get those crypts forming. And then those crypts go on to undergo fission and spread out, and you have clonal clonal interference that's happening and competition that's happening throughout the development of the colon. And then once you get to this point where you do begin to form a polyp, the idea is that we think that there's multiple seeds or crypts that are actually going into these polyps and forming these polyclonal initiation, at least at that initial stage. And so we built a mathematical model to examine this idea of mono versus polyclonal initiation. And this is just a non-spatial agent-based model. We use two different approaches where we consider it as a cell or as a deme or crypt. And we grow out a single pool of cells to about 10 to the 8. And we allow for mutations to accumulate within this. And then we sample from this either one seed, which is our monoclonal case, or 2, 5, or N seeds, being our polyclonal cases. And then we perform in silico sequencing on these polyps at the ends of the simulations. And so when we simulate this with several polyps with one seed, we never observe any subclonal sharing within any of these. We do see some clonal mutations or clonal ranges, which is just going to be those mutations that are present in kind of, we can consider it as patches or from the sampling being present in a larger number of cells. Once we get to 2 seeds, we begin to see some sharing of mutations, but still within that clonal range. And it's not until we get to about 5 seeds that we begin to see mutations that are shared within that subclonal range. And we can keep bumping this up to 10 seeds or 20 seeds. And it's really dependent on the number of seeds as well as the early mutation rate that's occurring as to how many you would observe. But as you get to too many seeds that are actually giving rise to these polyps, you have a theoretical maximum as to what the frequency of these mutations can exist at. So on the right there, I'm showing the simulation results with 5 seeds, and you see the kind of loss of number of mutations that are shared kind of dissipating as you move towards more polyps. And that's not quite the case when you increase those number of seeds and they exist at a much lower frequency. And this is what we're kind of seeing with the data, which is mirroring kind of the lower number of seeds. So this idea isn't new. There's been a lot of work since about 1960s all the way until most recently that's been looking at this. And yeah, I think it's an interesting phenomenon to try to understand precancer initiation. So yeah, I ran out of time, so there we go. Acknowledgements, yeah. My mentor, Christina Curtis, and Aziz Khan, who's a bioinformatics wizard, as well as all of Schenck Consortia. Thanks.
You know what's coming.
I don't. I thought I knew last time, but I don't.
Well, you know, crypts have got a very spatial architecture.
Yes.
And you completely ignored it.
Yeah.
How do you think that would impact your results?
I think that it would decrease the kind of the number of it would decrease our chances of being able to observe polyclonality or polyclonal initiation. At least that's what they found through one of the studies that I was highlighting there, was through the X inactivation, looking at some mosaicism within the colon and basically saying that polyclonal could only be observed if it's happening at the edges of those ranges, which actually biased the idea of monoclonal initiation. So I think it's important. I think that it decreases the number of clones that you can actually get.
Well, that's the thing, is that a lot of the published papers that have looked at this and developed spatial models show monoclonality, right?
Well, that's monoclonal conversion within a single crypt.
Yeah.
And so yes, that is true. It doesn't go against that at all. But what we're saying is that we think that there's actually kind of a community or small grouping of crypts that may actually be giving rise to these, to the precancerous hyperproliferation. Yeah.
Okay, thanks.
Yeah.
Very cool. Like, what do you think are the consequences of the polyclonal initiation? And is there evidence of polyclonal initiation in non-familial cases? Because look here, obviously, there's like lots of crypts that start going awry, but in more of a kind of health, non sort of normal wild type, whatever it means.
Yeah.
It's sort of unlikely that you would have multiple centers, but then there's this field conservation, so it's feasible, potentially possible. So what are your thoughts on the consequence of this polyclonal origin?
So I think the consequences is, and this is one thing that I actually skipped, is that we don't have good tools for understanding cancers that emerge from a polyclonal origin. A lot of the bioinformatics tools, especially, are they assume monoclonal origin. So this is our kind of affects our ability to resolve these clusters, the origins of heterogeneity. I think that a lot of this is happening as a transient state, because in our dysplastic samples, I wish I could have shown you this, we see that we do move towards a clonal convergence in these polyps as they do go on to the adenocarcinoma. So that's just to answer the one question. I think it's important for early detection and possibly understanding early metastatic seeding, especially, and possibly resistance, but I don't know. It's not my area. But as far as evidence in other tissues, Barbara Parsons in 2008 did a really great job of kind of summarizing the evidence based off of X chromosome inactivation studies that show that there was pretty extensive evidence of polyclonal initiation across different cancer types and different tissue types. So yeah, it's quite interesting. Yeah, thank you.